Dear audience, today I would like to explain the lymphatic system and the lymphatic organ and its most important aspects. In the first part of the lecture, we'll define the most important basic terms such as the lymphatic fluid and the basics of the lymphatic circulation, and after that we will go in detail and we'll discuss the most important aspects of the lymphatic organs. The lymphatic system is a very important system of the human body. It is meant to protect us and fight against different pathogens, foreign bodies, and pathologic components of the body, such as tumor cells. The major components of the system are the lymphatic fluid, the lymphatic vessels, and the lymphatic organs. I'm pretty sure you are familiar with this chart, which shows us the basic principle behind the different filtration systems of the human body. Okay, the green line shows us the oncotic pressure, and the red line shows us the hydrostatic pressure. As long as the hydrostatic pressure is over the oncotic pressure, filtration happens. As soon as this ratio changes and the hydrostatic pressure goes under the oncotic pressure, reabsorption takes place. With this system, with this filtration, approximately 20 liters of extracellular fluid is being produced in the human body daily. You can see that this 20 liter is much more than the average volume of blood in the human body, so that suggests that we need a significant reabsorption, which happens, and approximately 80 to 90% of this fluid is being reabsorbed. However, that 20 to 10% of fluid which is not being reabsorbed will remain amongst the tissues. This means about 2 to 3 liters of fluid, which is a lot, and this becomes the lymphatic fluid. As you can imagine, the lymphatic fluid in composition is very similar to the blood plasma. The lymphatic vessels are marked here with green. Okay, you can see that they are starting blindly, and at the beginning, you can compare them to capillaries here. At the beginning, they are much larger in diameter. Their very important characteristic is the incomplete basal lamina and the discontinuous endothelial lining. This allows different structures such as proteins and lipids to pass through their walls. After picking up the extracellular fluid, the lymphatic system transports the lymphatic fluid towards, for instance, primary lymph nodes, secondary lymph nodes, different lymphatic organs. Please note that the largest lymphatic vessel in our body is in the posterior mediastinum. This is what we call the thoracic duct. Here you can see the histological representation of a lymph vessel. Okay, so you can note that the wall is resembling a capillary. However, you can compare it to other vessels. You can see that the diameter is significantly larger than seen in capillaries. You can put the lymphatic organs into two major groups. One group is the so-called primary lymphatic organs. What's the difference between primary lymphatic organs and secondary lymphatic organs is the fact that in primary lymphatic organs, the lymphatic cells are being produced. Here you can see the bone marrow. On the example of the bone marrow, we can more or less collect the most important features of lymphatic organs. It is characteristic for almost every single lymphatic organ that the basic structure is made by reticular connective tissue. And you can also, that here on this hematoxylinogen stained slide is not very well visible. However, you can see that the lymphatic organs are very cell rich, especially true for the primary lymphatic organs. This is not very surprising. Here on this chart, you can see the formation of the different blood cells and lymphatic cells. The whole process starts from pluripotent stem cells, and they can differentiate into various directions, and eventually they create the cellular elements of the blood and the lymphoid, first the lymphoid stem cells and eventually the different lymphatic cells. The second primary lymphatic organ is the so-called thymus. The thymus is sort of an exception from the primary lymphatic organs, since the basic framework is not reticular connective tissue but epithelial reticular tissue. What does that mean? At the very beginning of its development, the thymus has no lymphatic cells, only composed of epithelial cells, so that's why we call it the thymus epithelialis. Eventually, when the lymphatic cells migrate into the thymus, it becomes the lymphatic thymus or thymus lymphaticus. As you can see, this era is until roughly puberty. At puberty, the medulla and the cortex proportion is going to start to decrease, and with years it will decrease to almost an undetectable level, and the fat portion will increase. So that's how the thymus involution happens, and the thymus becomes the thymus adiposus. So in most of our adult lives, we have this organ. However, there is a fourth possibility, the thymus persistens. This is a sort of a pathological stage which can be caused, for instance, by the adrenal gland pathology. So this situation means that the thymus exists in a sort of functional form throughout the life. On this histological slide, you can see the thymus, which is not very well visible, that the thymus is surrounded by a capsule, okay, by a connective tissue capsule, mostly collagenous fibers and some elastic fibers. What you can see is that this capsule sends trabecles into the structure of the thymus. So this is how the thymus will have its lobules. Okay. In case of the lobules, if you examine them a little more closely, you can see that its cortex is more basophilic, so more darkly stained, and its medulla is more lighter colored. Okay? So in the cortex, that's where the multiplication of the T cells, of the preforms of the T cells happen, and as these T cells first multiply and then migrate towards the medulla, they will develop and they will undergo what we call positive and negative selection. Okay, this is a sort of quality control that they recognize the foreign as foreign, and they recognize the own structures as own structure, and they do not react to them. The reason why the cortex is a little more basophilic is that it's full of small lymphocytes, small naive lymphocytes, and there is a lower portion of the epithelial reticulum cells. And on the other hand, in the medulla, there is a relatively lower proportion of these T lymphocytes and a higher proportion of the reticular epithelial cells, and they are also larger. You can also find other cell types in the thymus, such as macrophages and interdigitating dendritic cells. Here we turn to the secondary lymphatic organs. What you can see on this slide is not an organ on its own, but it's a very important organizing complex of the lymphatic tissues. This is what we call the lymphatic follicles. Okay, lymphatic follicles can be found in many organs. I'm very sure that you have seen such structures in multiple different organs by now. There's two types of the lymphatic follicles. The first one are the primary follicles. Primary follicles are not very large, roughly 50 to 100 microns in diameter. They are what we call the naive forms of the lymphatic follicles. That means that these follicles weren't exposed to antigens yet. So that's why in order to find primary follicles in larger quantity, we need intrauterine tissue samples. Once the B cells, so the B lymphocytes, get exposed to antigens, they will trigger a germinative reaction, and primary follicles become secondary follicles, which are first and foremost larger in size, 200 to 400 microns, and they have this very specific organization, what you can see right here. You can see that the center is lighter in color, right here. This is what we call the germinative center. Basically what happens that the antigen-exposed B lymphocytes will activate and they become centroblasts, and they multiply, and they push the other original cells of the primary follicle to the side, and they will become the mantle or the cap of the follicle. Besides the B lymphocytes, the FDCs, or in other terms the follicular dendritic cells, which are antigen-presenting cells, are very important cells of the lymphatic follicles. There are also some T helper lymphocytes, and very importantly some macrophages as well. The second structure which we need to discuss amongst the secondary lymphatic organs are the lymph nodes. Lymph nodes are such structures which we can see macroscopically. Basically, they are responsible for immune reactions against antigens, and they are fulfilling sort of a filtering function in the lymphatic circulation. Their framework, just like the majority of the lymphatic tissues, is made of reticular connective tissue. What you can see on this schematic image, that they are surrounded by a capsule, and this capsule, similar to the thymus, sends its trabecules towards into the structure of the lymph node. After that, it's followed by a cortex. This cortex is marked by this light yellow color. Within the cortex, you can find follicles, which are B lymphocyte containing structures, and between the follicles, in the so-called interfollicular spaces, you can find T lymphocytes, okay, so in the interfollicular space. After the cortex and before the medulla, we have this orange colored structure which we call the paracortex. How you can distinguish cortex from paracortex is the fact that there are no follicles. And the paracortex is T lymphocyte heavy, and it contains interdigitating dendritic cells for antigen presentation. And this area, the so-called HEV or high endothelial venules, are very common, which are such venules which do not have a continuous endothelial and basal membrane lining, which allows lymphatic cells to enter and exit the circulation. And the innermost part of the lymph node is the so-called medulla. In the medulla, you can find great many B lymphocytes, and the medulla is organized into medullary cords, and between the medullary cords, you can find medullary sinuses. And eventually, here, where the blood vessels are entering and exiting the lymph node, here we have the hilum, or the hilar area. This image shows us the general organization of the circulation of the lymph nodes, because there are double circulation here, meaning that we have to distinguish the blood circulation, which resembles the normal blood circulation. Arteries enter, eventually they capillarize. Capillaries will collect into venules, and venules collect into larger veins, and they exist. Here we have to mention the HEVs, which are wider in diameter, and they allow the entry and exit of lymphatic cells to the circulation. The other type of circulation which we have to mention here is the lymphatic circulation. As you can see so this is the hilum. The other side, compared to the hilum, we call the antihilar side, and then the antihilar side, the afferent lymphatic vessels enter the lymph nodes. Here, the sketch shows us that there are these valves which make sure that the flow of the lymphatic fluid is possible only in one direction. Okay, so these afferent lymphatic vessels enter the subcortical or marginal sinuses, which can be seen right here. After that, the sinuses continue along the trabecles in the trabecular sinuses, and eventually they enter the paracortical sinuses, which leads us into the medullary sinuses. You can see that the medullary sinuses are wide pool-like structures, and they also have this continuous endothelium and lamina basalis. This allows also, just like the HEVs, bidirectional migration of lymphatic cells into the lymphatic fluid and out of the lymphatic fluid. And here we can find along the walls of the medullary sinus macrophages which stick their protrusions into the sinuses, and they work as mechanical filters. And eventually the medullary sinuses will collect into the efferent lymphatic vessels. Here we also have valves. And an interesting fact of the lymphatic fluid within the efferent lymph vessels is that they contain approximately 50 times higher number of lymphocytes than the afferent ones. The next organ we will have to discuss is the spleen itself. The spleen has two distinct functions. One is immunological, the other one is hematological. On this hematoxylin stain slide, you can see that the spleen is surrounded by a dense connective tissue capsule, which sends, this is not that well visible anymore, trabecles towards the parenchyma of the spleen. You can see that, you cannot see, but since this is a lymphatic organ as well, its framework is reticular connective tissue, and the parenchyma of the spleen can be divided into this darker colored areas and to the rest. Okay, so this here, the more basophilic areas are the so-called white pulp, and the rest we call the red pulp. What do these terms mean? First, I would like to define the meaning of the white pulp. The white pulp is basically the lymphatic part of the spleen. This makes up roughly 20% of the entire pulp mass. In this image, you can quite clearly see the finer structures of the spleen. You can see that the capsule itself sends trabecles into the structure of the spleen. Within these trabecles you can find the branches of the splenic artery, which are called the trabeculate arteries. These trabeculate arteries gives rise to smaller arteries which exit the trabecles and enter the structure of the parenchyma of the spleen. These arteries, which you can see right here, are surrounded with cell-rich cylindriform sheets. These are called the periarterial lymphoid sheets, or the pulse. These sheets are made up of lymphocytes, macrophages, and interdigitating dendritic cells. The pulse is mostly made up by T lymphocytes, so this gives rise to the majority of the T lymphocytes of the spleen. The reason why the central artery is called central arteries is because they run in the center of the pulse. As you can see right here, follicles, mostly secondary follicles, are connected to the pulse, and they make up the B cell content of the spleen. It's not quite clear on this image, but more or less you can see right here that between the white pulp and the structure of the red pulp there is an approximately 80 to 100 micron thick so-called marginal zone. On this side of the white pulp you can find quite small, approximately 5 to 10 micron thick so-called marginal sinuses. In these marginal sinuses there is a possibility for the so-called homing mechanism of the lymphoid cells, meaning that they can enter or exit the bloodstream in the direction of the structure of the spleen. So basically they are the splenic equivalents of the high endothelial venules. If we take a higher power magnification of this area of the spleen, then we will have the chance to better understand the blood flow inside of the spleen. The central arteries themselves branch up to smaller arterioles and eventually capillaries, and they have several different ways to fulfill their function. A group of the capillaries fulfill a so-called nutritive function, which means that the arterioles capillarize and the capillaries will collect into venules, and the blood will be led back to the venous circulation system. In these capillaries, there is no possibility for the lymphoid cells to cross the wall of the vessels. In another group, the terminal arterioles will enter the mentioned marginal sinuses for the purpose of homing. And probably the third is the most interesting ones. So the central arterioles will branch further in a brush-like form, and these arteries are called the penicilliform arteries. The capillaries from the penicilliform arteries, at least a majority of them, will be surrounded by reticular cells and macrophages, and this is the this is the area where the macrophages will be able to reach into the blood vessels and filter out the antigens. And the following part of the circulation is a very unique very unique part of the circulation, because here in the spleen there is one of the few examples for the open circulation. So basically, from the arterioles, the capillaries arise, and the capillaries end freely within the trabecules of the spleen. The trabecules of the spleen has a reticular connective tissue framework, and on this reticular connective tissue we can find macrophages, plasma cells, granulocytes, and thrombocytes, but a low number of lymphocytes as well. So basically, the most important structure within the red pulp are the pulp sinuses. These sinuses look just like this one here. They are elongated, and their diameter is approximately 40 micron. The endothelial cells are also unique, since they are elongated, spindle-shaped, and as you can see right here, they do not create a continuous surface, which combined with the fact that the basal membrane is also perforated, they make it possible for cells to pass through the walls of the sinuses. So the capillaries which arise from the different branches of the central artery will dump their blood content openly in amongst the trabecules of the spleen, and they roam there freely with the purpose of filtering out the old red blood cells. Eventually, through the holes on the wall of the sinuses, the cellular elements of the blood will be able to migrate back into the blood circulation and eventually get back into the normal closed circulation. Some of the capillaries actually do not dump their blood load openly into the structure of the spleen, but they directly flow into the sinuses, and they will be led back to the the blood will be led back to the venous part of the circulation system. Since the lymphatic system is a very important line of defense to us, so it's not a big surprise that we can find lymphatic organizations on the front lines, so basically on such areas of the human body which have direct connection to the surface. Such organizations are within mucous membranes, for instance, Mucosa-associated lymphatic tissue, or abbreviated as MALT. A few examples for MALT are in the intestines, gut-associated lymphatic tissue, GALT. These are the Peyerplatches, bare patches right here, or in the airways, the bronchus-associated lymphatic tissue or BALT. This is what you can see right here. And obviously they can be found within the skin, named skin-associated lymphatic tissue or SALT. On the body surfaces there are such structures which are very well known, and they are lymphatic organs. They are the so-called tonsils. There are different tonsils in our body, and they can be distinguished based on their surface epithelial organizations and their content, if they contain muscles, in which directions, and if they contain cartilage or not. Let me just give you one example, which is probably the best known tonsil, the palatine tonsil. The palatine tonsil can be found in the so-called isthmus faucium, which is basically the border between oral cavity and the pharynx. The epithelial surface is stratified squamous non-keratinizing epithelium, as you can see right here. Underneath the epithelium, you can see it's very rich in secondary lymphatic follicles within the lamina propria. Please note these crypts right here on the surface of the palatine tonsils. Yes. This image shows a real photograph of the isthmus faucium. Here in the middle you find the uvula, and right here you would find the tonsillar fossa. As you can see, these white structures are sticking out from the crypts, since in the crypts broken down tissue material, pathogens and other structures can fill up and form these crypts, and this is a very well-known pathology which we call the follicular tonsillitis. So this is when pus accumulates in the crypts of the palatine tonsil. And with that being said, I would like to say thank you for your kind attention, and I would like to point out this very nice cartoon called Once Upon a Time. This explains the functions of the human body very well on a cartoon level. Thank you so much.